To sequence DNA by the chain termination method, the DNA must first be obtained in single-stranded form. A mixture containing the single-stranded DNA, DNA polymerase, the 4 deoxyribonucleotides A, T, G, and C, and a single short primer is prepared. The primer has a nucleotide sequence that is complementary to the 3' end of the region to be copied, and is required so that DNA polymerase can initiate DNA replication. This reaction mixture are placed in each of 4 tubes, and a different dideoxyribonucleotide is added to each tube. When a deoxyribonucleotide is inserted into the growing chain, replication continues. However, when a dideoxyribonucleotide is inserted, strand synthesis is terminated. The contents of the reaction tubes are then transferred to 4 lanes of an electrophoresis gel, and the oligonucleotides are separated by size and nucleotide type. The shortest oligonucleotide moves furthest down the gel. Reading from bottom to top, 1 base at a time, provides the correct DNA sequence.